Myself, Dr. Prashant Kumar, Associate Professor, Department of Biochemistry, C. Siddhartha Medical College. So today's topic is glycolysis. Definition of glycolysis: it is a sequence of reaction converting glucose into pyruvate or lactate with the production of ATP. So glycolysis is nothing but breakdown of glucose molecule into either pyruvate or lactate, and along with that there will be a production of ATP will be there, and NADH will be there. That NADH will enters into the electron transport chain later. So glucose is a sweet, and it is also called, glycolysis is also called as a embedded Mayer pathway. The glycolysis takes place in cytosol. So glucose will enter into the cell later. There are two types are there. Glucose utilization takes place is insulin dependent, that is in skeletal muscle, adipose tissue and cardiac muscle it will take place, whereas insulin independent, that is in RBC, brain and liver it takes place. So coming to the glycolysis, it takes place in three phases. So energy investment phase, splitting phase and energy generation phase. It includes 10 reactions, or you can divide it into two phases like preparatory phase and generation phase, that is 5 reactions. Next, coming to the first one, energy investment phase, where glucose is phosphorylated to a glucose 6 phosphate enzyme. It is catalyzed by the enzyme hexokinase, which splits that ATP into ADP, and it is the glucose is phosphorylated at the 6th position, glucose 6 phosphate. So hexokinase is a key glycolytic enzyme. The first step is glucose to glucose 6 phosphate. It is catalyzed by the hexokinase or glucokinase. Both are isoenzymes. Hexokinase present in all the cells, whereas glucokinase present in only liver. The difference between glucokinase and hexokinase is, glucokinase it is specific for glucose and it phosphorylates it, whereas hexokinase it acts on all the 6 carbon containing, hexose means all the 6 carbon containing sugar units, whereas glucokinase it has a high Km value for glucose and therefore it has a low affinity to the glucose, whereas hexokinase has a low Km value for glucose, hence it has a high affinity towards the glucose. This glucokinase is inducible enzyme. It is induced by the high levels of insulin and glucose, whereas hexokinase is a non-inducible enzyme and it operates it all the times, and glucokinase is not allosterically inhibited and it is not inhibited by glucose 6 phosphate, whereas hexokinase is allosterically inhibited. It is called as a feedback inhibition. Whenever there is a more amount of glucose 6 phosphate is there, if it is not utilized, it will inhibit the hexokinase. Glucokinase present mainly in the liver and some extent pancreatic beta cells, whereas hexokinase present in all the tissues, liver and extra hepatic tissues. So first step is glucose is converted to glucose 6 phosphate, which is impermeable to the cell membrane. It is a central molecule with the variety of metabolic fates. So this glucose 6 phosphate, either it may enter into the glycolysis or glycogenesis, gluconeogenesis or hexose monophosphate shunt. This is the first step. Glucose is converted to glucose 6 phosphate by the enzyme hexokinase or glucokinase. Here one ATP is utilized and hexokinase require magnesium here. The glucose is phosphorylated at the 6th position here. So glucose is converted to glucose 6 phosphate. Next, glucose 6 phosphate is converted to fructose 6 phosphate by the enzyme phosphohexose isomerase. Here glucose and fructose are isomers. So here isomerization takes place. So glucose 6 phosphate is converted to fructose 6 phosphate. Here general this one structural this one will be same, only there will be a spatial arrangement takes place here. So, glucose 6 phosphate is converted to fructose 6 phosphate by the enzyme phosphohexose isomerase. Next, that fructose 6 phosphate is phosphorylated to fructose 1,6 bisphosphate by a enzyme is phosphofructokinase, which is a irreversible and it is a regulatory step. Here you can see that fructose 6 phosphate is converted to fructose 1,6 bisphosphate. Bisphosphate by the this one is phosphofructokinase is the enzyme. Again here one more ATP is utilized. Next, second phase, that is splitting phase, where 6 carbon compound fructose 1,6 bisphosphate is splits into 2 3 carbon compounds, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate by the enzyme aldolase is the enzyme which splits that 6 carbon into 3 3 carbon compounds, and the enzyme is a phosphotriose isomerase which catalyzes the reversible interaction between glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Thus, this dihydroxyacetone phosphate is converted to glyceraldehyde 3 phosphate. From here onwards, there will be 2 molecules of glyceraldehyde 3 phosphate will undergoes glycolysis. Here next third phase is energy generation phase, where 2 molecules of glyceraldehyde 3 phosphate is enters. Here enzyme is glyceraldehyde 3 phosphate dehydrogenase which converts that glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate. It is here both oxidation, this one will takes place here, oxidation and phosphorylation. Here NADH is produced which is a high energy compound, and iodoacetate and acinate are the inhibitors here. This NADH will enters into the electron transport chain. There it produces 2.5 ATPs are synthesized. So, here ATP production is depending on the shuttle pathways. Here you can see that reaction. Glyceraldehyde 3 phosphate is converted to 1,3 bisphosphoglycerate. Bis means here 2 separate phosphorylation, this one phosphate groups are attached, that is at the carbon number 1 and carbon number 3 here, which is catalyzed by the glyceraldehyde 3 phosphate dehydrogenase enzyme. 1 NADH is produced here. Here both oxidation and phosphorylation takes place. Next step is 1,3 bisphosphoglycerate is converted to 3 phosphoglycerate. Here enzyme is phosphoglycerate kinase. Here one ATP phosphate groups is released here. So, enzyme phosphoglycerate kinase acts on 1,3 bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3 phosphoglycerate. Whenever the enzyme ends with kinase, either there will be a production of ATP or ATP is utilized there. So, it is called as a substrate level phosphorylation, since ATP is synthesized from the substrate without involvement of electron transport chain, and reaction is reversible. Next, 3 phosphoglycerate is converted to 2 phosphoglycerate by a enzyme phosphoglycerate mutase, which is an isomerization reaction. Here you can see that the phosphate group which is attached at the third carbon will shift to the second carbon to form a 2 phosphoglycerate. Enzyme is phosphoglycerate mutase. Next, 2 phosphoglycerate is converted to phosphoenol pyruvate. So, enzyme is enolase. Here water molecule is removed. Here you can remember that phosphoenol pyruvate, the enzyme will be enolase. That enzyme reaction, that is 2 phosphoglycerate by the enzyme enolase. Enolase requires Mg plus and Mn this one, and it is inhibited by the fluoride. Hence, for the collection of blood glucose estimation in the lab, the fluoride is added to the blood to prevent the glycolysis by cells. So fluoride is a competitive inhibitor, and enzyme pyruvate kinase catalyzes transfer of phosphate from phosphoenol pyruvate to form a ATP here, which is a irreversible step. Here you can see that phosphoenol pyruvate is converted to pyruvate, which is a end product of oxidative glycolysis. Here pyruvate kinase is the enzyme, ATP is produced, which is also irreversible step. So once again I will summarize that this is a glycolysis. Here glucose converted into glucose 6-phosphate, where one ATP is utilized, which is catalyzed by glucokinase or hexokinase enzyme here, and which is a irreversible step. And glucose 6-phosphate is isomerized to fructose 6-phosphate by the phosphoglucose isomerase enzyme here. Next, fructose 6-phosphate is converted to fructose 1,6-bisphosphate by enzyme phosphofructokinase. Here one more ATP is utilized. Next, this is the energy investment phase. Then the splitting phase is here 6-carbon compound is splits into 3-carbon compounds, and both are here triose isomerase can convert that dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Later glycolysis continues with the 2 molecule of glyceraldehyde 3-phosphate here. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, which is oxidative phosphorylation takes place here. Next, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. 1 ATP is produced here. Next, 3-phosphoglycerate is converted to 2-phosphoglycerate by the phosphoglycerate mutase enzyme. Next, 2-phosphoglycerate is converted to phosphoenol pyruvate by the enzyme enolase. Here one water molecule is released, and last step is again it is irreversible. This one, phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase. One more ATP is utilized here. So under aerobic aerobic conditions, pyruvate is reduced by NADH to form a lactate in presence of lactate dehydrogenase enzyme, which occurs in tissues lacking of mitochondria like RBC, lens and cornea, also in skeletal muscles during the intensive workouts. Here the NADH which is utilized here is obtained from the reaction catalyzed by the glyceraldehyde 3-phosphate dehydrogenase enzyme. So glycolysis will proceeds even in the absence of oxygen to supply a ATP. Under anaerobic condition, this pyruvate is converted to lactate by the enzyme lactate dehydrogenase. Here NADH is utilized. Coming to the energetics, under aerobic glycolysis, end product is pyruvate. Under anaerobic glycolysis, this one lactate is the end product. Here this glyceraldehyde 3-phosphate dehydrogenase, which produces 2 NADH here. So 2.5 into 2, total 5 ATPs are produced. That NADH will enters into the electron transport chain to produce a 5 ATP, whereas enzyme catalyzed by the phosphoglycerate kinase, where the substrate level phosphorylation takes place, there 2 ATPs are produced, and pyruvate kinase again 2 ATPs are produced. So total ATPs are produced here is 9. 5 plus this one 2 and 2, 9 ATPs, but in the energy investment phase 2 ATPs are utilized. If you minus that, the total ATP produced in the aerobic condition is 7, whereas in anaerobic condition 2 ATPs are produced.